Hello, and welcome back. What we're going to cover in this video now are the specific stages of mitosis. Okay, I've got over here just a reminder to show you that cell cycle. Remember, the cell cycle, the bulk of the time is spent in interphase, and that interphase is classified by G1, S, and G2. Okay, mitosis is the shortest portion of that part of the cell cycle. All right, mitosis is divided into stages as well, and that's what I'm going to discuss over here on this part of the board. Okay, mitosis, first step that we're going to get to is going to be prophase. All right, and then we're going to have prometaphase. I'm going to draw what the difference is between prophase and prometaphase, but in reality, if you're looking through a microscope, you're not going to be able to distinguish well between prophase and prometaphase. All right, so I kind of group them together. And if you use a much older textbook, like when I was in school, this was considered prophase 1 and prophase 2. Okay, so there are some differences and we'll discuss them. I kind of group them together. Okay, just keep in mind your lab instructor may not, so that's why I'm going to put these up here. Okay, from prometaphase we go to metaphase, metaphase to anaphase, anaphase to telophase, and then finally cytokinesis. Okay, this is another, these last two are also ones that you'll sometimes see grouped together in textbooks. Some will combine telophase and cytokinesis. I'll talk a little bit about what the differences are. Okay, but we're going to start out, we've got our cell up here that's in interphase. Okay, you can tell a cell is in interphase because you can see a clear nuclear envelope. You can see the nucleolus inside of the nucleus. The DNA is very thin and strand-light. We refer to that as chromatin. Okay. You're also going to have one set of centrosomes. This is a structure we haven't talked about yet. Okay. But those centrosomes are going to be the structure in the cell where the microtubules are going to originate from. Okay. And that's going to become important because the microtubules are going to be what are going to attach to our chromosomes and physically move them to the two separate cells. Okay? So in prophase, what we have happen, all right, our nuclear envelope starts to disappear. Our nucleolus is completely gone, all right? And what's going to happen inside the nuclear area there, you're going to start to see chromosomes condensing. And I'm going to just draw 4 here for simplicity's sake. Be aware when we're talking about human cells, there are more than 4 chromosomes. Okay? What you also have happen in prophase, let's see, our chromosomes condense, so you start being able to see them as chromosomes. And our centrosomes, or centrioles, are going to replicate. All right? So we've now got 2 copies of our centrosomes. And what's going to happen, those centrosomes are going to move to opposite poles, opposite sides of the cell. Okay? Let me draw the arrow in so that we know our directions here. We're going to go from interphase to prophase, from prophase to prometaphase. Okay? Prometaphase now, what's going to happen? Those centrioles have migrated to the poles. Okay, we've got microtubules coming off of there. And what's going to happen in the center here, we've got our chromosomes. what's going to happen, there's going to be a specialized structure on our sister chromatids here that's going to occur at the centromere. And I'm drawing that in pink there, okay? That's going to be our kinetochore, okay? The kinetochore now is going to be where our microtubules are going to start to attach, okay? And what'll happen, all right, those microtubules are going to attach to the kinetochore. And in pro-metaphase, those microtubules are kind of going back and forth to get those chromosomes lined up in the middle of the cell, okay? So with pro-metaphase, we completely lose the nuclear envelope. Our centrioles are at opposite poles or opposite ends of the cell. We can see our chromosomes. The microtubules start to attach to the kinetochore, all right? And we're going to start moving those chromosomes into position, all right? With metaphase, got our centrioles again. Now what happens? Our, excuse me, our chromosomes are going to line up in the middle of the cell. We've still got our kinetochores there in the middle with that bit of pink, okay? And what's going to happen, we've got microtubules that are going to attach to each of the kinetochores. And essentially, how that cell finds the middle is those microtubules are going to be of equal length, okay? So what we've got there, and I'm going to draw this as a dotted line, because really this is an imaginary line. It's not an actual structure in the cell. We've got what's known as the metaphase plate, okay? So that's just kind of an imaginary line that goes across the middle of the cell, and that's how our chromosomes are going to line up. And eventually what's going to happen, those sister chromatids are going to separate, and they're going to move to opposite poles, okay? So we went interphase to prophase, prophase to prometaphase, prometaphase to metaphase. Metaphase now is going to go into anaphase. And this is the really dynamic part of mitosis. And I have a video embedded in the PowerPoints for you to look at. I think it's really neat. But what happens then during anaphase, we've still got our kinetochore here, but what we're doing now is we're shortening those microtubules. That pulls apart the sister chromatids, okay? And we're now going to segregate one set of chromosomes to one side of the cell, one set to the other, okay? Now what's important is these two ends of the cell are genetically identical, okay? With telophase, what we have happen, we've still got those centrioles or centrosomes, okay? We're going to have our nuclear envelope reform. Our chromosomes now are going to start to decondense. They're going to kind of be thin fibers again. We'll get our nucleolus to come back. Let me label that nuclear membrane. And what happens, and what I've got here that's unique, that's a little bit different from all the other cells that I drew that were round, is we have this indentation that happens, okay? That indentation is going to be our cleavage furrow, okay? So what's going to happen, actin molecules are going to kind of pull on that cell. that cleavage furrow is going to come closer and closer together until we have two separate cells, okay? That process of cytokinesis then is the generation of the two separate cells, okay? Hopefully these made sense. I've got a couple other videos for you to watch that are not me talking. So make sure that you check on those. If you have any questions, please let me know.